Okay, so we're going to continue our discussion of synapses today. And we're going to move from understanding just one single synapse to zooming out a little bit and thinking about how we have multiple neurons working together. Okay? A couple review pieces from last time. So here's a practical example of something we might be applying some of what we've learned about in so far. So here's an example where we have a neurotransmitter, serotonin, that is going to bind to a channel receptor called 5-HT3. And when it does, that's going to allow sodium to cross a cell membrane. Okay? So the last time we learned about postsynaptic potentials, our excitatory and inhibitory postsynaptic potentials. So our test first is going to be to determine, is this going to create an excitatory postsynaptic potential or an inhibitory postsynaptic potential in our next cell, in our next neuron, in that postsynaptic. Okay, let's take a look. Okay, great. Yeah, so this is going to create an excitatory postsynaptic potential, an EPSP, in our postsynaptic cell, right? So we know that we have a strong driving force for sodium to come into cells, to come into our neuron. So if serotonin acting on this channel linked receptor, telling it to open and let sodium in, this is going to excite the next cell. This is going to create a graded potential that leads us closer to that threshold of negative 55 millivolts. Okay. So last time we talked about both inhibitory and excitatory postsynaptic potentials. So we can describe synapses themselves as either excitatory or inhibitory, which would be linked up to what neurotransmitter is there and what type of receptor is it acting on. Does it open something? If it opens something, do positive charges come in? Do negative charges come in? Do positive charges go out? Or are we blocking something from moving across? Right. So when we see inhibition, this usually has to do with potassium and chloride channels because potassium wants to come in. And one of the actions of chloride, but it also wants to come in, causing inhibition. Sometimes it stabilizes membrane potentials as well. Okay. We know that we got a kind of flow chart as we're thinking through our postsynaptic potential, right? So we know that the way that this is happening is we have our presynaptic neuron, action potentials are coming down that presynaptic neuron. We're firing more action potentials from that first cell, that presynaptic neuron. We're going to end up with more neurotransmitter in our synapse. It's going to bind to a receptor, might open a channel, might ultimately close a channel. We're going to think about what that does to the membrane potential. So does it make our cell more negative? Does it make our cell more positive? Okay. We can think about that because really what this is doing when we have a receptor, either directly or indirectly get us to opening or closing a channel, we're changing the permeability of that membrane to specific ions, which means we're changing how many of that ion move, either leading us to a dip, membrane potential going down, creating hyperpolarization, or a little spike, a depolarization if our membrane potential goes up. So our excitatory synapses work the same way as our inhibitory synapses. So we're going to see in our easiest examples, channels that open and let sodium and calcium in, or other ways that we can get this cell excited, get it membrane potential up is maybe we block potassium from leaving. So if we close potassium channels, this is another way we can start to inch up towards threshold. Okay. So in the case of opening a sodium channel, that's about moving sodium in. In the case of closing a potassium channel, we're going to expect some sodium to keep moving. Potassium isn't leaving to balance it out. So overall, the net effect is going to be that membrane potential going up a little bit. We also named our types of receptors. So just as a quick review of that bit. So we mentioned this 5-HT3 channel receptor that serotonin can activate. So our other ways of naming our receptors were ionotropic and metabotropic. And we want to make sure that we can link up these terms. So if we directly open a channel, when we have a neurotransmitter binds into that channel as a ligand, what's the name for that? The other way we refer to it. This is not working. No G protein here. Just channel link. So directly linked to the channel. Oh, okay. Yeah. So when we see something described as a channel receptor or a channel linked receptor, we are going to call those ionotropic receptors, directly opening and closing the ion channel. In our metabotropic receptors, we have an association with additional membrane protein, particularly our G proteins, right? So here's an example. In this case, the stabilization bit on chloride, but here's an example of a channel-linked receptor, an ionotropic receptor, right? So we have just channels where a neurotransmitter can bind in, right? So this is the type of situation we have when serotonin acts on that specific receptor, the 5-HT3 receptor. It does interact with other types of receptors as well. So these are ionotropic here. And if we wanted to see a G protein coupled receptor, those would be our metabotropic receptors. I don't think I have an image popping up, so I'm just going to draw one. Right, so here's our cell membrane. Similarly, we'd have a receptor, right, that could receive a neurotransmitter. Right, so we'd see the same binding of a neurotransmitter. Right, but instead of having that protein itself be a channel, we'd be linked up to a G protein that breaks apart when that neurotransmitter binds in and then activates potentially a channel pretty directly. So that would be our faster form of our metabotropic receptors called direct coupling, or we might even have an additional step. We have our membrane, we have our receptor, we have our neurotransmitter that has to bind in, we have our G protein, okay? But when that G protein breaks up, scoots to the side, it's not acting on a channel immediately, it's acting on some kind of enzyme that then makes stuff, some type of second messenger. Okay. And then that second messenger ultimately is what helps us open some kind of channel. right? So this is why our G protein coupled receptors work slower, but also why they can have longer lasting effects, right? In this example of our slowest type of metabotropic receptors. Metabotropic. Okay. Right. The slowest type, that second messenger, it's going to hang around in the cell even after that neurotransmitter leaves. It's not going to exit the cell immediately. It's not going to get broken down immediately. So we might have a long-lasting effect on this postsynaptic cell if it keeps that ion channel open for longer, even after we have the neurotransmitter wash out of our synaptic cleft. Okay. So now we're going to think about integration and convergence. Okay. So a reminder about our ion channel, right? The types we've had so far. We're thinking about cell locations, neuron locations right now. We had our leaf channels throughout our neurons everywhere, right? So our parts of the neuron, we had dendrites, right? Kind of the hair part of our neuron. We're thinking of it as a cartoon character. We have the soma, so this body. We have an axon hillock as we start to pinch off into the long skinny axon. And then we're going to have some number of axon terminals at the end of the axon. Right? So leak channel, you're going to have it everywhere. Right? That's what's setting our resting membrane potential at -70 millivolts. When we think about our, I'm going to skip one, right, our voltage gated channels, we've talked about the fact that those are really important for sending off our action potential. So we see them specifically concentrated in our axon hillock and in the axon, right? Got them everywhere, but these are major places we need to remember that we have those voltage gated channels. Now we know that our special third type of voltage gated channel, the calcium one, is over here in the axon terminal, right? So these are really locations we'd have the sodium ones and the potassium ones. Okay. Now that we think about our ion channels, our ligand gated channels from this list were the one thing that we hadn't really talked about when we saw this slide before. Right? We saw this slide a couple of times before. So we now know that those ligand gated channels, those are really receptors, right? So those are really receiving signals. Those are receiving neurotransmitters, other types of chemical signals. So that's why we see them in our areas that are going to be receiving signals. Right? So those ligand gated channels would be like on the dendrites and on the soma particularly. Because that's the part that's going to be touching, right, the synapse. If we're thinking about that postsynaptic neuron, right? So we got our chain. So these are where we have our different types of ion channels. Now we kind of understand they're all for different purposes. So as we think about neural integration, we're going to be thinking about that system where we have a presynaptic neuron, we have a postsynaptic neuron, but we're going to be thinking about when we end up having more complex systems of neuronal communication, especially in our brain, than just this kind of one-to-one relationship. So one thing we want to realize, right, we already know, if we have an action potential triggering, if we have our membrane getting to threshold of -55 from its resting potential of -70, that's going to trigger an action potential. And particularly, we're thinking about depolarization happening at the axon hillock first, then spreading down towards our axon terminal ultimately. Okay. If we have a postsynaptic potential that changes our membrane potential a little bit, but doesn't hit threshold, that makes no difference to our postsynaptic neuron. No action potential is going to get propagated down our axon in that case, right? So especially if we have inhibitory postsynaptic potentials, we're not going to have an action potential in the next cell. But also if we have just a little bit of excitatory action, doesn't hit threshold, no dice. So our neuron is going to have to figure out what to do when we have situations like this. Okay. So it's easy-ish enough when you have just one neuron, presynaptic neuron, talking to a second neuron or postsynaptic neuron. But in our actual brain and in our, basically everywhere we have neurons throughout our body, we have more connections than that, right? Learning is about building these connections. So we may see situations, we do see situations where we have a single presynaptic neuron, right? And then many different postsynaptic neurons that we're communicating with, right? So we can see axon terminals on this neuron. Wait, let's give them letters A, B, C, D. All right, so we've got these 4 different postsynaptic neurons here. All right, so what's going to happen when we take an action potential coming down this cell, right? It's going to end up sending that message to all 4 of these postsynaptic neurons. So we're going to refer to this as divergence, right? So when we diverge, we're spreading out, right? So that first presynaptic neuron has something important to say, wants to say it to a lot of different people. It's saying to all these many different postsynaptic neurons in divergence. And then the complicated one for us, where we have to think about, okay, how are things going to sum up? How do we add things together? Is the opposite. So convergence, right? So coming together, we're converging. In this case, we see lots of different presynaptic neurons here, right? If we're zooming out and not focusing on just one synapse, right? We've got communication from a lot of different places, okay? And any one of these might be eliciting some excitation, right? We might be letting some sodium in, or we might be sending a neurotransmitter in saying, hey, no, I want you to let more potassium out, or I want you to block potassium, right? We're going to be getting potentially conflicting signals from these different presynaptic neurons that are all converging on a single postsynaptic neuron. So that's convergence. And that's what leads us into thinking about summation. So when we talked about summation before, we were thinking about graded potentials, and we were thinking about the fact that they can be added together either in space, so our spatial summation, or in time, our temporal summation, so getting the same signal multiple times in a row. Now we're going to be thinking about that a little more concretely, thinking about specific neurons, specific arrangement of neurons. We're going to be thinking about things like facilitation of this signaling. We'll talk about what that means in a minute. Okay. So when we're thinking summation and when we're thinking about convergence, right? So multiple signals converging on this one postsynaptic neuron, right? We're thinking about what do we do when we're adding together some excitatory postsynaptic potentials, right? So we see those in green, but maybe we're also adding in some inhibitory postsynaptic potential, right? So we have multiple synapses here. We have communication with multiple other neurons, but ultimately we just either fire off an action potential or we don't, right? So we're thinking about how does this all add together when we get to the axon hillock. Okay. So we literally just see how they stack together, right? So here we see what happens if A fires a couple times. Right, so we're picking out A. Your book uses green for excitatory and red for inhibitory. Right, so this one's green, A is green. This is excitatory. So we can see if it fires once, a little bit of excitement, doesn't get us to threshold. We fire multiple times, but kind of slowly. we've got enough time for that little bit of excitation, that little depolarization to fade away in between. But if we have A fire twice close together, we jump up once, we jump up again, we get to threshold. So that's our temporal summation. That's what that would look like here. All right, so A fires off twice. Neurotransmitters end up in our synaptic cleft, trigger our receptors, they wash away if we're thinking about those first two slow firings. But if we're thinking about firing close together, we're blasting this synaptic cleft with lots of neurotransmitter, washes away slower, so we have this bigger effect, this temporal summation. In the second image, we see our spatial summation. So here we can see that both A and B cause slight depolarizations. Neither is enough to actually trigger an action potential. But we can see that if A and B fire at the same time, they do depolarize us enough to trigger our action potential. And it's not just about the fact that we had two presynaptic neurons firing together, because if instead of firing together A and B, we took A and C, right, we can see that C creates a little bit of hyperpolarization here, and we got this dip. So if we were to fire A and C at the same time, that's multiple signals being added together. This is still spatial summation, but this actually leaves us with a flat line membrane potential. They just canceled each other out. So in order to get to threshold, we got to be adding together the right signals, right? We've got to be adding together multiple excitatory ones. So when we think about how we can send stronger signals or weaker signals and how we can really fully affect whether a postsynaptic neuron fires off its own action potential or not, we're going to see that the way we do that is by changing the frequency of action potentials. Right? We know we can't make one action potential bigger or smaller than another. All we can do is vary how fast they come together. Right? So if we have a lot of depolarization happening, happens at the axon hillock, gets us to threshold, we fire off one action potential down the line. We stay depolarizing and sending depolarizing signals to the axon hillock. We're going to end up firing more action potentials after that refractory period is over. So more action potentials are going to result in more neurotransmitter, which likely is going to give us a greater effect on a postsynaptic neuron. Whether that's inhibitory or excitatory is going to depend on the receptor and the neurotransmitter themselves, and what they are. We dial in on presynaptic modulation. So here in presynaptic modulation, we're thinking about an additional step of kind of regulation of it. So far, we've just thought about, we send off action potential, we receive neurotransmitters, maybe we create an action potential, we hit threshold, maybe we don't if this is inhibitory or if our signaling is too weak. We've mentioned that we can have multiple presynaptic neurons converging on a postsynaptic neuron, and we have to do the summation piece to figure out, do we end up at a big enough membrane potential change? Now we're thinking about, okay, so beyond thinking about how a presynaptic neuron and a postsynaptic neuron are linked together, we now can have these modulating neurons that change the effect a presynaptic neuron has on the postsynaptic neuron. So we said that most synapses, right, are axodendritic or axosomatic when we're thinking about presynaptic, postsynaptic neurons, right, because they're either coming into the dendrites or coming into the soma. Here is where we have our exceptions. When we're thinking about these modulating neurons, you can see this modulating neuron here is coming into the axon of that presynaptic neuron. Right? So here we have an axo-axonic connection where our modulating neuron comes in and touches the axon, or really the axon terminal of that presynaptic neuron before our presynaptic neuron gets to have its own synapse with our postsynaptic neuron. So when we think modulating, we should be thinking about changing the effect, right? So we have two directions we can kind of change the effect of that presynaptic neuron in. We can either facilitate, right? So presynaptic facilitation is going to cause more release of neurotransmitter. And so presynaptic facilitation is going to make it easier for us to release more neurotransmitter here from that presynaptic neuron. Presynaptic inhibition is going to make it harder. We have presynaptic facilitation, easier, presynaptic inhibition, harder. So what's that going to look like? Messy. Okay. So we're going to start on the left side with our presynaptic facilitation. Right, so we're going to start here, our facilitation. Okay. So here we're looking at 3 different neurons and how they come in and affect a postsynaptic neuron, right? So between D and X, we can see that's just a presynaptic neuron and its postsynaptic neuron, right? So that's its own individual synapse, right? But if we focus in on the relationship of C and E, you can see that C is coming in and synapsing on X. But if we look at what E is doing, E is actually not affecting X directly, right? E is linking up to that axon terminal of neuron C. Okay. So what the images to the side are showing us next, right, is we're looking at what happens if just C fires. That arrow coming from the top is an action potential. So just C firing. This is a little bit of neurotransmitter. But in this case, to the right, we're looking at what happens if both C and E fire at the same time. All right, so you can see 2 black arrows coming down. All right, so here's an action potential coming down C. Here's an action potential coming down E. So you can see that when C and E fire at the same time, we end up with more neurotransmitter released. So that's why this is facilitation, right? So facilitation is all about the fact that we made it easier to release more neurotransmitter, right? More. Okay. So we can see what the effect of that is on the membrane potential down below as well, right? So C firing on its own gives us a slight EPSP, a slight excitatory postsynaptic potential, a slight depolarization, all good ways of describing this, right? If D fires on its own, same deal. So C and D working individually, firing off just one at a time, are roughly equal in size. If E fires alone, it does absolutely nothing, right? Because E is not affecting this postsynaptic neuron directly, not attached to neuron X, which is what we're measuring right now. It's only if we have both C and E firing at the same time that we have this magnified effect on our depolarization, actually getting us up to threshold. If we had both B and C fire at the same time, we'd want to think spatial summation, so that would likely get us higher up as well. But if D and E fire at the same time, E has no effect directly on F, has no effect whatsoever on C. So D and E fire at the same time, just looks like D's firing by itself. In our presynaptic inhibition, what we want to know is that it gets harder to fire, or it gets harder to release your neurotransmitter. Right, so what we're going to keep in on here, that when F and H fire at the same time, this is giving us less neurotransmitter. Right, so that's what we want to notice here. F and H are our two neurons that are linked up together in this modulating situation, right? So here we see F as a presynaptic neuron acting on Y, and then H is our modulating neuron acting on F out to the side. All right, so usually F is an excitatory neuron, usually creating a depolarization that we can see down here. Right, we can see that usually it fires off and releases a decent amount of neurotransmitter. But if H fires at the same time, the reason it's red on here is that this is an inhibitory effect. Right, so this is showing that when H fires, this gives us less neurotransmitter, which is why if F and H fire at the same time, we don't see that depolarization that we did when F fired as well. Okay. I've said it, but I want you to key in on something I see sometimes as a common misconception here. Usually when we see this inhibition, we're thinking about the postsynaptic potential, right? So we usually associate inhibition with things like hyperpolarization. But I want you to notice here that inhibition is actually about the amount of neurotransmitter when it's in this phrase presynaptic inhibition. So this doesn't necessarily tell you about the postsynaptic potential of that yellow neuron X or Y, right? All it tells you is about how much neurotransmitter C or F are going to fire. The direction of change is going to be dependent on whether C or F are themselves excitatory or inhibitory neurotransmitters. Okay. So let's have a written example of one. So we're going to imagine sequence consists of one presynaptic neuron, numbered neuron 1, one postsynaptic neuron, numbered neuron number 2. We're going to be thinking that modulator neuron X here with an axo-axonic connection is going to increase how much neurotransmitter neuron 1 releases. Okay, great. So we can use a question like this, which is a bit chunky, to check that we can draw this out, test our vocab. Right, so we know here, we say we've got presynaptic neuron 1. Right, we've got something like this. Postsynaptic means neuron 2 is next. Modulator neuron means we need something off to the side. In this case, we're seeing axo-axonic connection on neuron 1. So we're going to see something like this for X. Okay. And in this case, we're saying that when X fires at the same time, as neuron 1, we release extra neurotransmitter, which means this is presynaptic facilitation. Now that neurotransmitter could itself be excitatory or inhibitory. Happens to be inhibitory. This will still create an inhibitory postsynaptic potential on neuron 2, even though we have this facilitation happening between neuron 1 and this modulating neuron X. So focus on more neurotransmitter, less neurotransmitter for these words. So running through our types of synapses, just in writing, when we see these axo-axonic connections, we're thinking about the selective effect of a modulator neuron on one of our presynaptic neurons acting on one synapse. All right, so this is going to be kind of a local effect just to that neuron it's modulating, and just at the end when it starts releasing neurotransmitter, if we have an effect on the dendrites or the soma, so if we have a neuron coming in from its axon terminal signaling to a dendrite, signaling to the cell body, this is going to excite or inhibit the whole neuron that comes after that. So after one of those axodendritic or axosomatic connections, we're going to see our postsynaptic neuron either fully excited or inhibited, not selecting kind of what endpoints, which axon terminals that goes to. That excitation is going to spread all the way down to the end of the cell. If we have multiple axon terminals, it's going to end up affecting all of them because it affects the neuron as a whole. So here we have a summary, another different picture of what a neuron looks like. So we want to make sure that we can name the parts of a neuron and that we can see what's happening here. All right, so we can see what dendrites look like. We know the soma is just the cell body. Here we can see the axon hillock. Here we see the axon. In this case, we can tell we're looking at a myelinated neuron because we can see these chunks of myelin wrapping around with the nodes of Ranvier, these gaps in between. Here we see our many axon terminals releasing neurotransmitters. Okay. So we know in terms of information flow that what has happened is we've gotten action potentials coming down an axon, getting to an axon terminal where we trigger the release of neurotransmitter by affecting these calcium channels, these voltage gated calcium channels that let calcium in and trigger the release of that neurotransmitter from our vesicles. We know that in our postsynaptic neuron, we link up to some type of a channel. In this case, we're taking our simplest option and saying we're connecting to a ligand-gated channel, so one of those ionotropic receptors. In this case, we're thinking about how that might create an EPSP by letting sodium in, and then eventually leading to an action potential in that postsynaptic neuron. Our final topic is going to be some general anatomy related to our central nervous system, or our CNS. Okay. So in the future, we're going to see lots and lots about different branches of the nervous system and different ways of breaking it down. So what we're doing right now, we're thinking about the nervous system and breaking it down between what's housed by our axial skeleton. So you talked about the skull in lab, you talked about the spinal cord and the vertebrae in lab, right? So that's going to be your axial skeleton. It's our central nervous system, is that central part down the center line of your body. Okay. Well, that's one way of dividing our nervous system, right? So we're separating out here, central nervous system, or CNS, brain and spinal cord. And then all the nerves that come out of the central nervous system, so coming out to the sides, your different spinal nerves, making their way out of the vertebrae, right, would be going into our peripheral nervous system, so our PNS. So a nerve that you see like in your arm is going to be part of the peripheral nervous system. The nerve you see up in your brain are part of the central nervous system. In the future, we'll be talking about lots of divisions of the peripheral nervous system, as you can see on the chart, but we don't have to focus on that yet. Because when we're thinking about central nervous system, we're thinking about information processing. So the most sort of separation we have is we have some incoming nerves carrying sensory information in an afferent division, and we have some information going out in this efferent direction, so sending out our motor. Okay. So we don't really have to think about much branching when we think about central nervous system. So cells in the central nervous system, in the brain and spinal cord, are going to be a little different than the cells we see when we get out to nerves in the rest of our body, in the peripheral parts of the nervous system. Okay. So similarity they'll have between both of them is that we can conduct action potentials, so we can conduct impulses. But in the central nervous system, in the brain and spinal cord, you're going to see that our nerves generally don't divide, right? Okay. In general, nerve cells aren't going to divide if they're neurons, but we'll see that the central nervous system has these glial cells. These glial cells are neuroglia, right? Nervous system and glia for these glial cells. These are going to be cells that do divide, do replicate, generally support function of the neurons, but don't actually themselves conduct impulses. Okay. So we've talked extensively about neurons now and how they work. So we're going to take a peek at what's going on when we think about this nervous system more broadly, including those glial cells. Our brain and spinal cord are the main parts of our central nervous system. They help us maintain homeostasis, right? They're taking in signals about did your temperature change, did your blood pH change, etc., and helping us fix that by affecting effector organs. We've got about 100 billion neurons here in our central nervous system and about 100 trillion synapses. So lots and lots of cross communication. So the neurons are sending their action potential, right? Lots and lots of these neurons synapsing lots and lots of places. But the glial cells are just going to support this. Okay, so glial cells will end up being surrounding cells. Glial cells. If you wanted to separate up neurons more than we have done, one thing people do is describe them differently by shape. Another way people can describe them differently is by describing what they do. So they might sense stuff, sensory neuron. They might send out action potentials that affect a muscle or a gland. Those are going to be our motor neurons, those efferent ones. Sorry, there's a lot of like duplicate terminology. So I'm just trying to make sure that I try and link those words as often as possible. So these are going to be our afferent nerves and our efferent nerves later on. That's good to start linking those up even now. Okay. And then we also see some neurons that are fully within the central nervous system. Don't leave it, don't talk to the peripheral nervous system at all. So we might have some association or interneurons in the central nervous system itself. Now we have the glia, sorry guys. So you'd think the 100 trillion synapses, those 100 billion neurons in the central nervous system would be the main part, but they're actually not. They require a lot of support. And actually about 75 to 90% of your central nervous system are these support cells, the neuroglia. I've tried to condense them down into some bite-sized little functions so that we get some just general familiarity with these terms, but they do a lot more complicated stuff than this. Okay. So we're going to see a couple different types of glial cells. Right, our astrocytes Astro, think astronomy. They're kind of star-shaped. They're these big green ones. Okay, so astrocytes. One of their super important functions is going to create a barrier between our brain and our blood. So you can see here in this image how the astrocytes, one of the things they're doing, right, they're grabbing onto neurons, but they're also holding this capillary kind of closed, covering over any gap in it. So this is going to regulate what kinds of things can cross into your brain, actually into the tissue, right? So like what medications can actually come in, what components of your blood can actually signal to the brain. Another type of support cell we'll see are the cells that make that cushioning fluid around your brain, that cerebrospinal fluid that gets generated by our ventricles. So the cells that are doing that are our ependymal cells. Okay. So think like epithelium is your skin lining the outside of your body. Ependymal cells are lining that ventricle, lining this cavity. Our microglia, think of those as like our immune system for our brain and spinal cord. So phagocytosis is when you eat another cell. So our microglia are going to be swimming around, helping defend the central nervous system from pathogens. And we have two types of cells that form myelin. If we're in the brain and the spinal cord, so our central nervous system, it's going to be an oligodendrocyte. So like grabbing onto dendrites is kind of how I would think about that, although that's not physically how they always look. They're these little octopus guys. Those are oligodendrocytes. So they make myelin on multiple different neurons, potentially. And our Schwann cells are also going to make myelin, but they're going to fully just wrap around one single axon, and that's going to happen in the peripheral nervous system. Okay. So we will stop there. We got a couple more slides that we can get through on Wednesday as we do our review, but I tried to summarize them all in that one slide anyway. I've also opened up the practice exam for this unit if you want to get started on that now that we've got all our content down.